Sathyabama Institute of Science and Technology.
Dear students, this is Dr. S.M. Indhumathi, Assistant Professor from the Department of Biotechnology, School of Bio and Chemical Engineering, Sathyabama Institute of Science and Technology. Today I am here for a presentation on the topic Lactose Operon, Structure and Regulation. Introduction. So operon is basically a structure which contains linked genes that codes for different proteins. In case of lactose operon, this operon is usually seen in prokaryote. Prokaryotic gene regulation has various operons like lactose operon, tryptophan operon, arabinose operon and so on. So in case of lactose operon, which is seen in Escherichia coli, it is a cluster of three structural genes which encode certain proteins that are involved in lactose metabolism, and this lactose operon speaks about certain set of genes in the DNA that regulates the operon as well. So many protein coding genes are seen in bacteria in clusters which serves as transcriptional units. So the first model of operon was actually described to the world by Jacob and Monod in the year 1961. Usually, when an organism, for any organism for that matter, we need energy source to survive. So this lactose operon speaks about whether the organism is relying on glucose as for its energy source or lactose for its energy source. In the presence of glucose, lactose operon remains inactive because any organism will actually prefer a basic carbohydrate, a monosaccharide which will be easy to break. Rather, if in the absence of glucose, an organism prefers to take lactose. This lactose is basically a disaccharide and it has to be metabolized obviously. So it has to be broken into glucose and galactose and then further into the energy ATP. So this is a complex process, and this lactose metabolism requires three enzymes to be produced by the bacteria itself, and the production of these three enzymes by the bacteria is actually explained in lactose operon. So the structure of Lac Operon, you can see the diagrammatic representation of Lac Operon. The operon model actually proposes the following list of elements. The first are the three structural genes. You can see LacZ, LacY and LacA. Followed by that, we have a promoter site called PLAC, and this PLAC is actually bound by a cap protein called the catabolite activator protein, which actually makes a complex with the cyclic AMP. So this cap, which makes a complex with cyclic AMP, forms a final product called CRP, that's called CAMP receptor protein, which is actually the activator of the Lac Operon, and this particular compound binds to the promoter region of the operon, and we have an operator region here, and that is called as OLAC, and this operator region is the one which is recognized by the repressor protein, and the repressor protein binds to it to stop the transcription of those three structural genes. Actually, those three structural genes LacZ, LacY and LacA actually produces the three proteins or the three enzymes. So LacZ produces beta galactosidase, LacY produces galactoside permease, and LacA produces this thiogalactoside transacetylase. The function of beta galactosidase is to break the lactose into glucose and galactose. Galactoside permease, as the name indicates, it actually transfers the lactose into the cell, and this the thiogalactoside transacetylase actually detoxifies the final non-metabolizable products of lactose metabolism. Talking about the regulation of the operon, we have two types of regulation here, the positive as well as the negative regulation. So first we'll look into the negative regulation. In the negative regulation, we have the template DNA strand. So we already know what is transcription, translation and all those. We have seen it in our previous videos. So we already know that there is a template strand and a non-template strand, and the template strand runs from 3' to 5' region. So as you all can see, this is a template strand which runs from 3' to 5', and from the promoter region is the Lac Operon actually. And before the Lac Operon, we have something else called the regulatory gene or the Lac-I. I stands for inhibition of Lac Operon. So this regulatory gene has a particular sequence called Lac-I, which actually codes for a protein called the repressor protein. This Lac-I gene gets actually transcribed with the help of RNA polymerases, and the transcribed repressor mRNA will be translated to form the final repressor protein. And if this repressor protein is formed, this repressor protein will actually find the operator region in the lactose operon and it will bind to it. Thereby, the RNA polymerase which is bound to the promoter region of the lactose operon will actually be blocked. It will not be transcribing the 3 structural genes and there won't be any enzyme formation. And this is negative regulation, where the repressor proteins finds the operator, binds to it and blocks the RNA polymerase by transcribing those 3 structural genes forming the enzymes. Coming to positive regulation. The 3 enzymes are not only produced by lactose operon, and it is usually present in meager amounts inside the bacteria. So during times when the bacteria is about to take lactose as its energy source, it uses the meager amounts of previously existing enzymes inside it to break the lactose into a compound called allolactose. So it takes the help of the beta galactosidase enzyme to break the lactose to form a small compound called allolactose, and that becomes the inducer of lactose operon here. So the smaller amounts of enzymes that are already present in the bacteria will actually help the bacteria to produce larger amount of enzymes. So in this positive regulation, after the formation of allolactose, which joins as an inducer here. So the regulation happens naturally. You have the regulatory gene, there is a Lac-I which will get transcribed to mRNA and which will get translated to the repressor protein also. But during positive regulation, we have the inducers called allolactose joining the reaction. When this inducer joins the repressor protein, it makes conformational changes to the repressor protein, thereby inactivating it. An inactivated repressor protein cannot bind to the operator and it cannot stop the transcription of the 3 structural genes. As you all can see, the inducer has bound to the repressor and the repressor has become inactivated, and this repressor cannot find the operator and it cannot join to it. While the RNA polymerase, which was already attached to the P-Lac, this is the promoter of the Lac Operon, starts to transcribe the 3 structural genes to form a polycistronic mRNA. So we all know what is polycistronic mRNA, and mRNA which will actually code for different proteins. In actually eukaryotes, we have monocistronic mRNA. The entire mRNA will code for only one protein, but in case of prokaryotes, we have polycistronic mRNA which will actually code for 3 different proteins. In case of lactose operon, 3 different enzymes. So after the transcription of those 3 structural genes Lac-Z, Lac-Y and Lac-A, we have the polycistronic mRNA, and through the polycistronic mRNA, finally the 3 enzymes beta galactosidase, galactoside permease and thiogalactoside transacetylase are produced in larger amounts, which will actually help the bacteria to metabolize the lactose that is taken up as an energy source. So operon and glucose. The entire lactose operon is actually evolved on the basis of presence or absence of glucose, because glucose is the major inhibitor of Lac Operon, because glucose inhibits the enzyme adenyl cyclase, which is responsible for producing cyclic AMP. So when cyclic AMP is not produced, the catabolite activator protein cannot bind to the CAMP, and then it could not form the CRP and the operon cannot be activated. So the presence or absence of glucose will decide whether the operon will be active or not. So you can see that here, glucose on the other way, in its absence, it cannot inhibit this adenyl cyclase. So obviously CAMP will be produced, the cyclic AMP will be produced, CAP will be binding to the CAMP and it will produce the CRP and the operon will be active. So based on the presence of glucose and availability of or unavailability of lactose, the operon has evolved in different setup. So when low level of glucose is present and only lactose is available as a carbon source for the bacteria, the organism obviously has to depend on lactose as its energy source. So when there is low glucose, the organism takes up lactose from the medium for its energy and it tries to produce the enzymes through lactose operon. So the operon must be obviously active here. I mean, the lactose will be broken down into allolactose, inducers produced. Inducers will bind to repressors, which will be inactivated, and the repressors cannot bind to the operator, and the RNA polymerase that is bound to the promoter region of the operon will transcribe the 3 structural genes, thereby producing the enzymes. This is in case of low glucose presence. In case of the presence of high glucose and no lactose availability, the organism has no chance to take up lactose and the operon need not to be remain active. Rather, the organism simply relies on glucose. It takes up glucose for its energy and just survives. There is no presence of lactose here and no operon is needed, so the operon remains inactive. In case of low glucose and again lactose is unavailable, again there is no possibility of the organism to take up lactose. Even though there is very little amount of glucose present in the medium, the organism will solely rely on the lower amount of glucose for its energy and the operon again remains inactive as there is no presence of lactose here. Finally, if there is high glucose and lactose available, even now the organism, the prokaryotic bacteria will actually rely on the presence of high glucose. When there is glucose and lactose available, the organism will obviously prefer glucose. So it completely relies on high glucose, but if at all it needs more energy, if it needs to rely on the presence of this lactose, the operon will be slightly active. Rather than being more active, the operon becomes moderately active because the organism is now completely relying on glucose. Only when it is needed, the organism takes up lactose. So to metabolize such small amounts of lactose, the operon has to be moderately active to produce moderate amount of enzymes. So in this case, the operon remains moderately or slightly active. So this is all about lactose operon, the structure, regulation, and how this operon has evolved based on the presence or absence of glucose. So these are my references, and thank you students for watching the presentation.